Thank you. Thank you for the introduction. It's a pleasure to be back in Kansas City and to get to visit the Linda Hall Library. It's an amazing facility, and I had a great time here today so far. So so far, yeah, we'll see how the next hour goes. The topic of my talk tonight is human genomes, understanding ourselves genetically, using the genomes of our closest extinct ancestors, the Neanderthals and the Denisovans, which we'll get to in a minute. Before I get into this, I like to begin with acknowledgments, just so I don't forget, and so that you know that this was not just my work. There was a very, very large team involved in this, and here are the names and pictures of some of the people. The main guy here is Svante Pääbo, who was my boss when I was in Leipzig there, and still a colleague and a collaborator. He was the visionary who, back in the 80s, invented the field of ancient DNA, imagining that there was DNA, or could be DNA, in old material from animals who have been dead for hundreds or thousands of years, and working hard to figure out how to extract this and to sequence it. And some of the other very key people in this project who are population geneticists and biochemists, and Michael Lachman, just a crazy theoretician who's helpful in many ways. Okay, so understanding what makes humans humans, what makes humans unique, is one of the most interesting, compelling questions in all of biology and all of science, how we differ from the rest of the things in our living world. And people have been thinking about this for a long time. We now know that our closest living relatives are the great apes, the chimpanzee and the orangutan and the bonobo and the gorilla. We are most closely related to these, of all the things out there in the natural world that we see. These numbers here, you probably, if you think about it for a second, can guess what these are. This is an estimate of the number of individuals of each one of these species who are currently alive on the planet today. There are about 7 billion of us, and every one of the other great apes is critically endangered. If we were to think of our species really in ecological terms, that we are just one of many species on this planet, maybe that's not the right way to think about it, but if we were to allow ourselves to do this for a second and judge ourselves in ecological terms, we are really doing well ecologically. We're from the great ape ecological niche, we are doing well in this, and all the other guys are in deep, deep trouble. And I'd like to point out, as a just interesting thought experiment, that it wasn't that long ago. In fact, the history of this is traced out in the room back there and to the left. The primary articles that kind of trace this out, putting humans in the right context, saying where we fit in the natural world. This was done, the broad strokes of this came together just within the last few centuries. And these other guys, these great apes that are so similar to us, they're about the same size as us, you know, give or take a factor of 2. They don't have a tail. They have faces that are remarkably similar to us. These guys are on their way out. They may be extinct within the lifetimes of many people in this room. It's a good chance that some of them will be, except for zoo populations. So as we were learning, kind of putting it together, how we fit in the world, these guys are on the way out the door. How hard would it have been, how much harder would it have been if we were left only with monkeys, small things with tails, to try to figure out how we fit in the natural world? It already took us a long time having great apes that we can look at and see kind of face to face something that's not that different than us. It could have very well been the case that we didn't figure it out and only get to look at these little monkeys running around with tails and say, that might be something that's closely related to me. But in any case, the comparative approach, looking at things that are very closely related to us, the most closely related to us, chimpanzees, we can define what it means to be human to some extent. We can observe the chimpanzees very closely and observe humans and make long lists of things that differ between all humans and all chimpanzees. And these are the things that are most similar to us. These lists are, to some extent, an answer to this question, what makes us human? Because these are ways we differ from the things that are most similar to us. Here is just a subset of this list. The lists can be as long as you care to observe. You can look very closely at the fine structure of the hair and brain anatomy and all kinds of things, biology and physiology, and find lots and lots of differences between humans and chimpanzees, and these are the things that separate us. We know that every human who is born will be bipedal, will stand on 2 feet, will have much less hair compared to a chimpanzee, will have the capacity for spoken language. All of these human characteristics are what we call 100% heritable. We know that every human who is born will have this. So what we start life with, what everybody in this room started life with, if you go backwards in time through your life, when you were a child, when you were a toddler, when you were a baby, when you were 1 cell big, when you were 1 egg cell that was in your mother and it was fertilized by 1 sperm from your father, all you were at that time was some cytoplasm and 1 copy of your DNA sequence from mom and 1 copy from dad. Already at that time, it was possible to say that you would grow up and be bipedal, and you would have this capacity for language, and you would have less hair, and you had all these human characteristics. That means that the instructions for making these characteristics are written somewhere here, in what we all start life with. 2 copies of our DNA sequence, 1 from mom, 1 from dad. This is a picture of chromosomes, the physical molecule that holds DNA. And they're arranged from largest to smallest. You see there are 2 of every one, from chromosome 1, the biggest one, up here, down to the sex chromosomes, X and Y, here. This is the physical copy of the instructions for life that we all start with. One copy comes from mom, and 1 copy comes from dad. And they come together, and presto, changeo, bingo, there you are. You grow up and you become bipedal. Likewise, the chimpanzee, they have exactly this thing here, except for this chromosome number 2 is actually 2 separate chromosomes. Sometime in human history, those 2 chromosomes fuse together and exist as 1 chromosome in humans. Otherwise, chimpanzee is nearly identical. 99 bases out of 100 are the same. But in that 1% difference are the chimpanzee characteristics. Okay, so as a geneticist, it's a great time to be working on human evolution because we have complete genome sequences from thousands of humans and about 10 chimpanzees and the gorilla and the orangutan, all the great apes we have complete genome sequence from, and many of the other primates, many other old world and new world monkeys we have complete genomes from. So we can turn this question around and say what makes us unique as humans genetically and get a very precise answer. We can line up the 3 billion bases in the human genome and the 3 billion bases in the chimpanzee genome and see what's different. Where are we different from them? And we can put our finger on the 35 million or so places where humans are different from chimpanzees. And this is satisfying to some extent. We answer the question very precisely, what makes us different. There are a few places where the chromosome appears to invert a little bit, and there's 1 chromosome fusion, or chromosome 2 came together, but it still leaves one lacking a little bit to answering this big question. Where are the genetic differences that lead to these biological differences? Where are the genetic differences? Which of these 35 million genetic differences lead to bipedalism? Which of these lead to spoken language? How do the genetics make us this individual that's uniquely human with all the things that we care about biologically and physiologically? And that, it turns out, is very, very hard. These lists of genetic differences are very long, going into the millions, and the biological differences are as long as you care to make them. As much as you care to observe chimpanzees and humans, you can find more and more differences. So that's a hard problem. Is there any way then that we can make this easier? Can we make the lists shorter? And the answer is yes, we can. There are things that are more closely related to us than the chimpanzee. The only problem is they've all died, gone extinct. So this is some interpretation of the hominin fossil record. Hominins are all of the species that have lived since the time of the common ancestor of human and chimpanzee. These are they, from here in the past up until humans in the present. This, the field, paleoanthropology, people who look at the remains of hominins and try to figure out who's ancestral to who. It's a very contentious field. I was once told that there are more paleoanthropologists than there are actual bones to look at, which is a recipe for contention if there ever was one. So people argue a lot about whether Homo heidelbergensis and Homo erectus are actually the same thing, and Homo rhodesiensis, how is that related? There's a line that maybe didn't lead to anything, or maybe it did. But the broad strokes of this, everyone agrees on. There were Australopithecines, and then a transition to homoforms with a much bigger brain, and then there were the Neanderthals who went extinct, and there's human. And the Neanderthals, everyone would agree, are shown temporally here from left to right. The Neanderthals are our closest extinct relatives. The chimpanzee is our closest living relative. The Neanderthal is our closest extinct relative. Here is a picture of Neanderthal cranium, something that everyone would agree is a Neanderthal, classical Neanderthal. And shown on the bottom here is Cro-Magnon. This is an anatomically modern human. This is indistinguishable from a person who would be alive today, yet it's about 30,000 years old. This could have been your great-great-great-great times 2,000 grandfather. This is morphologically different. This differs because there's a brow ridge here and a occipital bun here. Neanderthals had lower heads and more slung back. A real expert, I am not, a real expert could tell you many, many differences in tooth morphology between Neanderthals and humans. And one can really get lost in the differences between the morphology of the Neanderthals and the morphology of anatomically modern humans. They must be different morphologically, or else we never would have recognized them as something distinct. They would just be old bones that someone dug up. These things that make them distinct led people to say this was something that doesn't exist anymore. It's distinct and now it's gone. But you can get lost in these differences, and I think it's helpful to put this in a little bit broader context. And if one looks at the cranium of our closest living relative, the chimpanzee, and compare the Neanderthal and human relative to this guy, the similarities actually start to come out again. It doesn't have this massive jaw or a very small brain case or these enormous teeth here. And the connections for muscle where the jaw connects are not massive like they are in the other great apes. So this guy is definitely different enough that you say it's something different, but is quite similar morphologically to humans. And we think Neanderthals were quite similar morphologically to humans. Okay, temporally, Neanderthals show up in the fossil record about 200,000 years ago. This is CO2 data from ice cores. Some folks have gone and drilled down a core into very, very, very old ice where snow accumulates every year and a little bit of gas gets trapped in there. And if you drill down in this, you can take from layers and measure how much CO2 is there. And you see that CO2 is thought to correlate very well with the temperature of the Earth. And there have been times in the past when it's warm, like it is now, and times where it's very cold, ice ages. People who talk about temperature change today, this is not really what they're talking about, climate change. This is massive climate change when huge swaths of the Earth were covered with glaciers, or times in the past when it was even much warmer than it is now. But Neanderthals, we know, lived through at least 2 ice ages, and they almost made it to the current interglacial time. So they show up about 200,000 years ago, and then they disappear from the fossil record about 30,000 years ago. We never find bones that are younger than about 30,000 years old that are clearly Neanderthal. Modern humans, anatomically modern humans, us, show up about 130,000 years ago, and of course we're still around till today. And what this means is that for most of the time that we've been around, and for most of the time that Neanderthals were around, we shared the planet. We were here together. And in fact, it wasn't just Neanderthals and humans that were around. There were late surviving erectus forms. There was a hominid that lives, whose bones were found on an island called Flores in Indonesia that stood about this tall and had a very, very small cranium. It was very small overall, but the cranium looks anatomically fairly modern. The wrists and the shoulder look very archaic, and people have no idea what this guy was and who he's really related to and how he got there. They call it the Hobbit because it's a little bitty person who lives far away. But that guy was dated to about 13,000 years ago. So if you go back about 40,000 years, the Earth is populated with many hominid forms that are quite different from us. Now, we're the only ones around. All these other guys have gone away, but it wasn't that long ago that that wasn't the case. Okay, so geographically, Neanderthals are principally known in Europe, but they also existed in the Middle East, and recently we've shown with DNA evidence that their range even extended as far as Siberia into Asia, and it's a bit of an open question now exactly how far to the east they extended. Were there Neanderthals, for example, in China? We would love to know that. But at least into Central Asia, Neanderthals wandered. The genome that we sequenced came primarily from these 3 bones. There were shards of long bones that were excavated in the Vindija cave in Croatia, excavated in 1980 and radiocarbon dated to about 38,000 years old. They spent 38,000 years in the, buried here in this cave, and then about 2 decades in a museum in Zagreb in Croatia. And now they are mostly ground up to take DNA out of them. Getting DNA out of bones is destructive sampling. One has to actually crush the bones and extract the DNA. So these have been sacrificed to the greater interest of science so that we can get the DNA. The way that we do this used to be, for a long time, a method called PCR that was invented by a speaker in this series who was here, I guess, last year, Carrie Mullis. He also went to my alma mater, UC Berkeley, and quite a colorful guy. When he and Svante Pääbo actually were coincident at Berkeley too, and when PCR was kind of being developed, one of the first applications of it was ancient DNA. PCR is this very powerful technology, stands for polymerase chain reaction. It allows one to amplify a small fragment of DNA into as many copies as you want. It's like a photocopier for DNA, but it doesn't work like a normal photocopier. It makes twice as many copies as you put in. So if you put in 2 copies, it'll give you 4. And if you put in 4, it'll give you 8. And if you put in 8, it'll give you 16. So you just run this over and over and over again and very quickly make as much DNA, exactly a copy of the starting input as you want. And this is great for ancient DNA because generally very little DNA is recovered from these bones. We don't have enough to do anything with. If you want to learn its sequence, you need to make many copies. So this was a great application of PCR, making many, many copies of something. But it's limited. PCR is limited for ancient DNA in that you have to define the segment of the genome that you would like to amplify before you do the PCR. So if you extract DNA out of a bone, you may have many millions of short fragments that came from the Neanderthal, but you have to say, I want this fragment, and only amplify that, and all the other millions are then lost. recently being 2005, there's technology developed that allows direct sequencing of everything that comes out of this extract. This technology was not built for ancient DNA, but it turns out it's perfect for it. And the idea is you take the DNA extract and you stitch on little bits of DNA onto the beginning and the end of everything, not knowing what everything is, all these millions of molecules. You put the same thing on the beginning and the end, and then you do PCR. PCR requires that you know a little bit of the beginning and a little bit of the end, and you amplify what's in the middle. Well, now everything has the same beginning and end, and what's in the middle is what you care about, and you just amplify everything and sequence it all together. This has really revolutionized the field of genetics and molecular biology. It has let us, for example, reduce the price of sequencing a genome from tens or hundreds of millions of dollars before to about $2,000 now. If you want your genome sequenced, if you want to know these complete 3 billion base pairs, the retail price of that as of today is about $2,000. And it's almost certainly going to get cheaper than that in the future. So we applied this, I applied this when I got to Leipzig to ancient DNA, and that's what we used to sequence the genome. The first things that were known about Neanderthal DNA were all centered on the mitochondrial genome. And there's a technical reason for this. The Neanderthal, or the mitochondrial genome, is a small circular genome. It's only about 6.5 thousand base pairs long compared to the 3 billion bases of the rest of the genome, and it exists in about 1,000 copies per cell, whereas the rest of the genome only exists in 2 copies per cell, the one you got from mom and the one that you got from dad. All 1,000 copies of the mitochondrial genome come from mom, but because there are so many copies around, it's easier to recover this. It's much easier to recover mitochondrial DNA, so that's what we knew about Neanderthals up until about 2005. Mitochondria, as I said, you get it from your mother. She also got it from her mother, who got it from her mother, and in this way, it traces one lineage through any person's, any individual's genealogy. You have the same mitochondria as your mother. My mitochondria, which I got from my mother, is an evolutionary dead end. I will pass it on to no one because I'm not female. Females pass on mitochondria, specifically to female descendants. They pass it on to their male descendants. Everybody needs this to live, but their male descendants won't pass it on to anybody. The nuclear genome, on the other hand, for every place in the genome, you're equally likely to come from any individual in your own personal genealogy. You're equal parts mom and dad, equal quarter parts your grandparents, eighth your great-grandparents, etc. Mitochondria, not so. 100% mom, who was 100% her mom, who was 100% her mom. Okay? But this is what we could trace in the Neanderthal up until a few years ago. And the relationship between Neanderthals and humans was very clear from this. Neanderthals in the mitochondria fall outside the variation that we see in humans today. Everybody who's alive today has a common ancestor here. This is oftentimes called mitochondrial Eve, the common ancestor of everyone in the mitochondria, has this common ancestor who was more recent than the common ancestor with Neanderthals. Neanderthals fall outside of our variation. And this is telling, in a way. It could have been, if humans and Neanderthals were interbreeding, that the mitochondria from Neanderthals would have come into humans. And you would occasionally today find a human who has a mitochondria that looks like what we see in Neanderthal. But that's not the case. And tens of thousands of people have had their mitochondrial genome sequenced, and we never see the Neanderthal mitochondria sequence in it. So this is kind of where the field stood for a long time. We could do a little bit of genetics, look at this one place in the genome, the mitochondria, and there's no evidence for admixture, for interbreeding there. Okay, and then the new generation of sequencers. This was the first one that we used, manufactured by a company in Connecticut, and this field moves very fast. They were the darlings of investors and the darlings of everything for a while. They were bought up by a Swiss company called Roche, and now they're a bit passé, and now everybody uses this machine, which is made by a company in San Diego, the Illumina sequencer. And there are rumors now about the next thing that will come along. And from my perspective, I love it, because every time there's a next thing, it's so much cheaper and so much faster and better, and it couldn't be better. It's bad news if you hang your hat on one of these companies, but it's great news if you're a consumer of these things. So in any case, these are the machines that we use to sequence the Neanderthal genome. And for the first time, instead of amplifying some small fragment, we just sequenced everything that came out of the bones. And we saw, somewhat to our surprise, that most of what we were getting, most of the sequences that were coming out of the bones, weren't Neanderthal at all. If we compare these short little DNA sequences to every sequence we've ever seen before, everything in the public database, most of the things looked like some kind of soil-living bacteria. It was DNA from microbes that have colonized this bone while it was sitting in the dirt for 40,000 years, which maybe we should have guessed, but we didn't know that until we sequenced it. And in fact, most of those looked different enough from any soil-living bacteria that we'd ever seen that we couldn't really even say what kind it was. So the vast majority of the sequence data that we generated in the Neanderthal genome sequencing project was not Neanderthal DNA. It was this extraneous DNA from bacteria that had colonized the bone. But a few % of the sequences that we generated were Neanderthal. They looked like primate DNA, and they were useful for us for assembling the genome. Okay. One interesting note, the Neanderthal DNA, the hominid DNA, if we look at how long the sequence fragments were, they varied. They were on average a little over 50 nucleotides each, but the microbial sequences were also quite short, and the version of sequencer that we use for this could read out to about 250. It couldn't go much longer than that, so if the sequence was longer, we couldn't see it. It would have to stop there. But that was plenty long enough, even for most of the microbial sequence, which would indicate that some of this microbial sequence, maybe a lot of it even, is also very old. Not only is it bacteria that colonized this bone, but maybe it colonized the bone a long, long time ago and describes microbes that are tens of thousands of years old. And we've thought from time to time, whenever someone gets a free minute, of doing more analysis of that. But we haven't. We've been focused on the Neanderthal sequence for the last few years. So as I said, there's lots of mitochondria. That's why we would PCR amplify it. It also allowed us to accumulate sequence in the mitochondria very fast, and we were able to assemble the complete 16.5 thousand base pairs of the mitochondria to get finished quality sequence. And that process is kind of shown schematically here. Every row is another sequence read, or some fragment of the sequence read. And at the top is what we infer the sequence to be. And each color is a different base. So green is A, blue is C, black is G, and red is T. So this sequence says CAA, CAT, etc., like that. So we get these reads and we line them up, and then every column is a single position out of the 16.5 thousand in the mitochondria. And having so many copies, have such redundancy at every position, lets us see through the occasional error. So we know this position is really a C, despite the fact that this one guy said T. And having this really high coverage, we can infer what the correct sequence was back when this Neanderthal was walking around 38,000 years ago, and infer things about the gene changes and do the interesting analysis that we would like to do. We also, having this really high coverage of the mitochondria, we're able to see what kinds of changes exist, how often. And you can even see it a little bit in this slide. Oftentimes, when the real answer is C, blue, we see a red T. This comes from chemical damage in DNA. This is just a quantitative description of that. When the real thing was C, oftentimes we read it as T. Likewise, when it was G, oftentimes we read it as A. Every other error happened very, very low, kind of what we would expect from the machines that we were using for sequencing, but these were really high. And we know this is ancient DNA-associated chemical damage. C, the cytosine base, will spontaneously deaminate to uracil, lose this amino group here. And then the machinery that we use to read the DNA, when it sees this unnatural base, this doesn't normally occur in DNA, it will read this as a T. T is what it looks most like. So where this C was, we get deamination to U, and then we read this as a T. This process, spontaneous deamination of cytosine to uracil, it's happening all the time. It's a natural process. It happens in any kind of aqueous environment. It's happening in you right now. And it's a large driver of the mutations that we accumulate over our lifetime and often lead to disease and cancer and other things like this. This process that's happening in the background all the time, there are repair enzymes that can recognize this uracil and then go and fix this. And they're operating all the time when we're alive and fixing this for the most part. But as soon as one dies, the repair processes are turned off. They don't have the energy that they need to go, and this just starts accumulating more and more and more and more. So we see a lot of it when we sequence the DNA, and now we can quantitatively know how much to expect, so we can correctly infer what the Neanderthal sequence was when he was alive. Actually, when she was alive. All three of the bones were from female Neanderthals, it turned out. Okay, so we accumulated little over 1 billion bases from each of the 3 Vindia bones. We also got a little bit of sequence data from some other Neanderthals just to make sure that the bones from this cave look like other Neanderthals. And the first thing that we were really worried about when we got this data was the problem of human contamination, DNA from humans who have touched the bone. And this is a very difficult problem when thinking about Neanderthal DNA because humans and Neanderthals are so closely related. To give you some idea of this, I said human and chimpanzee are about 1% different. If you just take a random 100 nucleotides in your genome and compare it to the same 100 nucleotides in chimpanzee, on average, one position will be different. One of those A, C's, G's, or T's will be different from what they have. The other 99 will be exactly the same. Okay? So that's how similar humans are to chimpanzees. Neanderthals are about 10 times closer than chimpanzees. So on average, we would expect less than 1 difference in 100 base pairs. And the fragments that we have are much less than 100 nucleotides long. Typically, they're 50 or 60 nucleotides long. On top of that, we've got this base misincorporation that makes the overall error rate something like 2 or 3%. So we've got noise of about 2 or 3%, a signal of less than 1%. And how are we to ever know that what we're looking at is real human or Neanderthal DNA or human DNA from someone who has touched this bone and contaminated it? You, before you leave the room tonight, will leave many thousands of copies of your genome in this room. We shed cells all the time and hair, and within this are copies of our genome. All of the people who touch these bones can presumably leave contamination if they're not careful, if they don't wear gloves and treat things very carefully. We try to be very careful, but you never know. So we devised what's shown here, these strategies for telling whether it's contamination or not. The first one is based on the mitochondria. We had the complete mitochondrial sequence for this individual, for the 3 Vindia individuals that we had sequenced. So we know every position there. We also know the complete mitochondria of thousands of people who are alive today. So we can find the positions, and it turns out there are about 130 of them, where every human, no matter who you are, you differ from a Neanderthal, from this Neanderthal. So all of those positions, whenever we have a read that covers one of those diagnostic positions, we can say, does it have the Neanderthal base, or does it have the base that all humans have? Is it contamination, or is it Neanderthal? And from that, we had about 27,000 reads that covered these good diagnostic positions, and 26,957 were the Neanderthal base, and 73 were the human base. So from that, we can infer contamination of less than 1%. More than 99% of the sequence data, at least covering those positions, is Neanderthal DNA. We also used the fact that they were 3 female Neanderthals to test for contamination. Because they are female, female does not carry the Y chromosome. Y chromosome is like to males what the mitochondria is to females. It only exists in boys, and you only pass it on to your male descendants, and it strictly tracks the male lineage. You got it from your dad, who got it only from his dad, who got it only from his dad. The way we generally inherit last names, it tracks the last name. You could write your last name on your Y chromosome, and that's how it's passed down. Okay, so the Y chromosome, if we see any Y chromosome sequence in our data, it's got to be contamination, because these are girl Neanderthals and they shouldn't have any Y chromosome sequence at all. We found, of the millions of sequence fragments, we found 4 that were clearly Y chromosome, and from that we can estimate contamination from a male human of less than 1%. We also sequenced the genome of 5 people who are alive today to compare to the Neanderthal, and I'll tell you about that in just a second. But the first thing we did with it was the test for contamination, and we found places where all humans had the same base, had the same A, C, G, or T, and that differed from chimpanzees. So for example, all the humans have an A, and the chimpanzee has a C. So somebody changed in the past. Either the chimpanzee picked up the C and it used to be an A, or the humans picked up the A and it used to be a C. Then, of all those positions, we ask, where do 2 of the 3 Neanderthals agree with the chimpanzee? So if 2 of the 3 Neanderthals have the C and all 5 of the humans have an A, then that A probably came up since humans split from Neanderthal. So it's probably a diagnostic position that separates Neanderthals from human, because we see it one way 2 times out of 3 in Neanderthals one way, and 5 out of 5 the other way in humans. And then we test that 3rd Neanderthal and ask for that 3rd Neanderthal. Now this is the one we're going to try to measure contamination. Does it match the other 2, or does it match the humans? And we can switch out which is the 3rd Neanderthal that we're learning from the other 2 and testing this 3rd one. And from this, we estimate contamination of less than 1%. Almost every time, the 3rd Neanderthal matches the previous 2. So sorry to belabor this point. I realize this is kind of a boring part in the talk, but if you think this is boring, just wait till you see what's coming next. Just kidding. This is very important to know that it is really Neanderthal DNA that we're looking at and not human DNA. So once we have convinced ourselves of that, we can compare every place in the genome where we see something in chimp, something in human, and something in Neanderthal, and ask who is different. And the things where Neanderthal is different and chimp and human are the same, they likely changed here, and human different changed here, and the chimpanzee here. If we just look at what the difference is, what base changed to what base, if it's a change that's specific to the Neanderthal, it is dominated by G's changing to A's or C's changing to T's, which is what we know is associated with ancient DNA. This is ancient DNA sequencing error. These are not real evolutionary changes. They're things that happened while this bone was degrading in the ground. This is not real evolution. Real evolution, it turns out, looks like this. GA, AG, TC, CT, these are transitions, and we know that they happen much more often than these other classes of changes, which are called transversions. All of these pairs are chemically similar. Dissimilar. All of these pairs are chemically similar, so those changes happen more often. Same thing, these changes that are restricted to the chimpanzee. They happen about equally often and at an elevated rate compared to these. We can then count these now and say how many differences are there here that are specific to humans since we split from Neanderthals relative to how many differences in humans since we split from chimpanzee. So in other words, if humans and chimpanzees split 6.5 million years ago, what percentage of that time in the past did humans split from Neanderthals? And it turns out for each of the 3 bones, it's about 12.7%. So that would be about 800,000 years ago that humans split from Neanderthals. Just looking at the number of differences and making this assumption, as we do in molecular genetics, that these changes happen in a regular way over time, in a fairly clock-like way. If you wait 1 million years, you'll get half as many changes if you wait 2 million years. There's a molecular clock, and it just spontaneously changes over time. So with that assumption, humans and Neanderthal, the average difference in the genome is about 12.7% of what it is back to this common ancestor with chimpanzee. Okay, so that's interesting. We wanted to do this same comparison with 5 humans that we sequenced. So we collected data, because it's so cheap to do this now, from somebody who lives in France, West Africa, South Africa, East Asia, and Papua New Guinea to compare to the Neanderthal, and kind of do the exact same things with these humans as we do with the Neanderthal sequence. And as I just said, the Neanderthal is about 12.7% diverged from the human. But this 12.7% is just an average. If we look across the genome and ask how much is that locally, there's a wide variance in that. The Neanderthals are in yellow and orange and red here. And there's some places where this number goes up to 20 or 30%, where the human and Neanderthal are quite different, and other places where it's quite low. For all humans, they are more similar to another human, the reference human sequence that we're using for this, than any Neanderthal is, but there's also a variance here. And we know that this is the case. We know, in fact, I'll show you in a minute why this is the case, why 2 people are very different at some part in the genome and very similar at some other part of the genome. So there's this variance here. There's not just a specific number, but in general, the entire distribution is shifted deeper for the Neanderthals, as one might expect. What we didn't expect at all was the analysis that I am going to describe here. The reference genome, the reference human, is just a single sequence of a human that was published about 10 years ago that is a composite of DNA sequence from many different individuals. At the time that they started this project, they asked for donors of DNA to be sequenced and go into this, and went to great lengths to make sure that they were anonymous donors and that they stay anonymized. And for a long time, that was fine. The records were kept clean and nobody knew anything about them. But in the intervening years, we've learned enough about the variation that exists within people that they can be de-anonymized to a certain extent. It can be said that this region of the genome came from a donor who's likely of European ancestry, or of African ancestry, or of Asian ancestry, because the particular variance that they have at places where people vary happens to be the one that you see more often in Europe, or happens to be the one that you see more often in Africa. So you can go across this one reference sequence that we all use that's an example of a human genome and say this part is European ancestry, this part's African ancestry. And this is very useful to us. If we partition the genome in this way and say, is the Neanderthal, which is 12.7% overall, diverged from the human, does that change if I'm looking at the European parts or if I'm looking at the African parts? And it turns out that it does. It's lower here, green, when we're looking at the European parts, and higher when we're looking at the African parts, which is completely unexpected because Neanderthals split off from humans before there were Europeans or Africans or any of the groups that you could name today. Neanderthals should be equally distantly related to all of them because they split off earlier in time. So this was one of the first clues that there may be a closer genetic relationship between Neanderthals and at least Europeans. So we devised this very simple test. This was the brainchild of a colleague of mine, David Reich, a great population geneticist at Harvard. This very, very simple test where we took DNA sequence from 2 of our 5 individuals, a pairwise comparison, and found, for example, the places where the French guy differed from the West African, where the French guy had a G and the West African had a T. And do this across the whole genome, find all the places where those 2 happen to differ, and then ask, who does the Neanderthal match, the French guy or the West African? If Neanderthal is a clean out group to everybody, then it shouldn't matter who you have here or who you have here. It's always 50-50. The Neanderthal matches one half the time and the other half of the time. And it turns out that if you compare the 2 Africans, it is 50-50. And if you compare any 2 of the non-Africans, it's also 50-50. But any comparison between one of the Africans and one of the non-Africans, you had statistically very large increase of matching between the Neanderthal and the non-African. And it didn't even matter which non-African it was. Even the non-African from Papua New Guinea has this extra matching to Neanderthal. And Neanderthals were never in Papua New Guinea. We've since run this test with DNA from people from the Americas, Native Americans. And not only were Neanderthals not in the Americas, no people at all were in the Americas for 10,000 years after Neanderthals went extinct. There's no way they were here because nobody was here. Yet Native Americans have this excess allele matching to Neanderthals. They have this excess similarity to Neanderthals. So the model that we have for this is that when some human migrants left Africa about 70,000 years ago from the archaeological record, and first came into Neanderthal territory, first encountered the Neanderthals, they admixed with them. They bred with them. And this population then grew and expanded and colonized the rest of the world and brought with it the genes from Neanderthals that they picked up at this time that they admixed with Neanderthals, and eventually even came into the New World with these Neanderthal genes, such that the genome, the DNA that is carried around in people today who are of non-African ancestry, is about 1% to 4% from Neanderthals. Okay? So that's the way the story stood for a few months, and then we found another bone, even better preserved bone from this cave in Siberia called Denisova Cave. And I've heard this pronounced both ways to address that. The cave was named from a hermit who lived in there in the 1800s who happened to be called Dennis. And that's why they call it Denisova Cave. This cave is remarkable in that it has a nearly continuous record of hominid habitation for over 100,000 years. There have been people living into it. These Denisovans, Neanderthals, earlier forms, modern humans, even up until the 1800s when Dennis was there. This cave is a very, very nice place to live if you're in the region, evidently. And it also preserves DNA very nicely. We got DNA out of a little tip of a finger bone, and we're able to sequence about twofold coverage of its genome. It looks, when we rerun this analysis with an updated version of the human genome, now for the Neanderthal, instead of 12.7, we get 12.1. So that number changes a little bit. But the Denisova number is 11.9, almost exactly the same, meaning that Denisova and Neanderthal are about equally distantly related to humans. If we look and regionally even, it looks kind of the same. If we make a phylogenetic tree of humans that we've sequenced and the Denisova and some of the Neanderthal that we've sequenced, the Denisova is more closely related to the Neanderthals than it is to the human. But these branch lengths indicate how different two things are, how long they've been evolving separately. The Denisova is more distantly related to Neanderthals than any two humans who are alive today. So it's kind of hard to conceptualize what it was. It was more like a Neanderthal than we are, but it was more different than a Neanderthal than any two people who are alive today. And all we have to say about it morphologically is the tip of a finger bone. We can't really describe it very well that way. But we can describe it 9 ways to Sunday genetically. We know everything about it genetically. And for comparison, we sequence lots more humans now because this is getting cheaper and cheaper, and did this excess allele matching test and found, shockingly, that this Denisova, unlike the Neanderthal, where the story was the difference between Africans and non-Africans, it looks the same between Africans and most non-Africans. It only looks different when you look at samples from these islands east of the Wallace Line in Oceania here. Individuals whose ancestry traces there have about 8% contribution of their genomes from this Denisova individual, or a population related to this Denisova individual, and probably not that particular guy. But that cave is up here in Siberia. And today, people who live here have genetic contribution from a very old population whose range was at least up here, and we have no idea how wide it was. And if you have an idea how that happened, I would love to hear it, because we are really struggling to make some sense of this. Have done more population sampling and can see geographically in a little more detail where the signal exists. Papua New Guinea, if you that's where it's highest. If you rate that as 100%, it's equally high in Australia and high in lowland in New Guinea, and a little bit in the islands around here. But like I said, west of this Wallace line, you don't see it. East of the Wallace line, you do. How it got to be there and distributed the way we see it now is largely a mystery. One idea, not that I necessarily advocate, but one possible idea is that when humans migrated out of Africa, we know that they show up here very early. So some of them probably very quickly made it to the islands of Oceania. If these people were early enough, and if the Denisova range extended far to the south enough, then they could have met the Denisovans. They could have been there early enough before the Denisovans went extinct and carried with them their genes that they picked up. And then later, people would more slowly colonize mainland Asia and eventually come to colonize into these islands. But they would have been too late to interact with the Denisovans and not have this Denisova genetic signal. But that's a hand-wavy kind of story that is there to just describe data after we see it, and I'm not at all attached to that explanation. There may be others. And it will be fun to see how this story develops. Okay, so I just want to finish up with, excuse me, a few things that my group in Santa Cruz is now working on using DNA from Neanderthals. And here are my two students now, Sam Four, a very talented computer scientist, and Brandon Rice, a molecular biologist. What we are doing is using the Neanderthal genome to probe for positive selection, some kind of genetic change that may be important and interesting and useful to humans since we split from Neanderthals. And also to ask, of these genes that we got from Neanderthals, what was the implication of this? Was there anything that's biologically useful or important, or is it all just sequence that could have come from anyone and doesn't have much consequence? One thing that most people who aren't in this field don't necessarily know is that most of our genome, there's really good evidence that it doesn't do anything. The genes that we know of, the 20,000 or so genes, they only comprise about 2% of these 3 billion bases. The other 98%, if you just look at the rest of it, about 50% of that are a small class of repetitive sequences that just occur over and over and over in the genome. Just occur over, and it doesn't seem at all to matter to evolution if they change. They change in a very clock-like way over time. They exist in chimps and other great apes and other primates, and they've just accumulated as many differences as you would predict from how long these things have been separate. So most of the genome, it kind of doesn't matter where you got it or even if it's present at all. What we would like to know is the particular regions that came from Neanderthals, are they all this stuff that's just background schmutz, or are they actually something that matters? And we're working on that. So before I tell you how we do that, I just want to do a little primer in genetic inheritance in sexually reproducing diploids. That's a mouthful. Diploids means you get 2 copies of the genome. There's some forms of life that only have 1 copy of the genome. We're diploids, like most animals are. We get 1 from mom and 1 from dad. We actually have a phase in our life where we're not diploids, where we're haploids. We only have 1 copy. That's when we're sperm and we're eggs. It's a very short part of our lifespan, but it's part of our lifespan. There's a time when this entity that you think of yourself, it was actually 2 separate entities. You were a sperm and you were an egg. And then that came together and it made this brain that you can use to think about the fact that you used to be a sperm and an egg. But we do have this phase where we have 1 copy from dad and 1 copy from mom. So we get 1 copy of our genome, each of our chromosomes. We get 1 of them from dad and 1 from mom, unless it's the Y or the X or the mitochondria. But the rest of it, most of it, it's like this. And you probably knew that, but what you may not know is that this copy of the genome that you got from your dad, it didn't exist in your dad. And this copy that you got from your mom, it didn't exist in your mom. Instead, it was put together, especially for you, in the sperm or in the egg that would become you. And the way that it was put together from your dad, for example, he got one copy from his dad and one copy from his mom. What he will do when he makes the sperm that will become you is stitch those two together so that it's the beginning here from the one he got from his mom and the end here from the one that he got from his dad. And now there's this new single copy of this chromosome that didn't exist as a discrete thing in him, but now it exists in you. And likewise, you will do the same thing. If you have descendants, you will take the genome you got from mom and dad and put them together in a single chromosome. In this way, every generation is the physical embodiment, the coming together, not of the parents, but of the grandparents. The mother's mother and father are put together in a single chromosome. And the father's mother and father are put together in a single chromosome in that generation. And when you have your children, you will give a single chromosome, either the one you got from mom or the one you got to dad. You'll stick these together and give that to your children, and your parents then will become the grandparents of this generation. I think you get the idea. Okay, so if you where this happens, where this stitching together takes place, is random. It could be anywhere in the chromosome. You could get, like what's shown here, a little bit from your dad's mom, and a lot from your dad's dad here. Or it could have been the opposite. It could have been down here, and you get a lot from your dad's mom and a little bit from your dad's dad. So this is just random, where it happens in the chromosome. What this means then is that if you trace back some particular place in your genome, some gene that you have, trace it back through the generations, that tracing back through the generations is different depending where you are in the genome, what gene you're talking about. So if there's a gene here in this yellow bit, for example, that will make you have a big nose like me, and you got this gene from your dad's mom, if the mom is on bottom. That yellow chromosome came from your dad's mom, okay? And if that's the one that makes you have a big nose, then you're unlucky. And if you ask, where is the gene that came from your dad's mom's dad, right there, so dad's mom's dad, green one, I don't got any green right there. I didn't get anything. My dad's mom's dad right there gave me nothing. It's lost. We don't just accumulate DNA from the generations. We get half of what our mom had and half of what our dad had, but the other half is gone. It's not inherited in us. Maybe it's inherited in our brother, because this happens at different places, but not in us. So if we trace this yellow back, it comes from dad's mom. If we trace the green back, that came from dad's mom's dad. And likewise, every place in the genome has a different path back through your own personal genealogy. So you may be separated by 3 generations from your cousin, or you may be separated by 3,000 generations from your cousin, just depending where you look in the genome. At some gene, you may have a common ancestor 3 generations ago. Another one, if you didn't happen to both get back to that ancestor, then you've got to go way back. And maybe it's 3,000 generations. Okay, so that's why when we look to see how close Neanderthals are to human, or 2 humans are to each other, we see this wide range. Some places 2 humans will be very close because this path happens to be a short one. Some places it's very far, and there's a long path and lots of differences. Okay, so this is useful now because we can construct these genealogies, these trees, at every place in the genome across humans. We have lots and lots of data from human genomes, and we can look for, we know that mostly these trees, if we relate all of humans together, they look like this or like this, where NA is non-African and the dark blue A is African. Most of the trees that relate all of humanity look like this. If you look for the deepest split, it's almost always the case that you see Africans on both sides of this split. Sometimes one side of the split is only Africans. but almost always you see the deepest divergences only within Africa. There's a deep message about human genetic diversity here. If you care about human genetic diversity, there's really no reason to ever leave Africa. That's where human genetic diversity exists. The genetic diversity that's present outside of Africa is largely just a subset of what you see within Africa. Okay, so this is most of the genome, but it's cut off here. About 4.4% of the genome, by this measure, looks like this. It's largely the exception, where you have a deep divergence that splits non-Africans from everybody else. So a very different set of genes that only exists outside of Africa. It's very rare when this happens, but when we see this, if we then ask, well, what does the Neanderthal have? Almost every time it matches this deep non-African version. It's almost always the case that if you see something very different and it only exists outside of Africa, it's because it came from Neanderthals. So this turns out to be a very powerful way of finding Neanderthal admixture, to say exactly this region, this gene came from Neanderthals, and this version of the gene came from Neanderthals. And we're doing this trying to find regions of the genome that are important, that came from Neanderthals. This collaboration with Peter Parham and Laurent Abi-Rached at Stanford, we've looked at a region called the major histocompatibility complex. It's a set of genes that has to do with pathogen recognition in our immune system. And what we've found is that these three genes, HLA-A, C, and B, the reason that we looked here is because we have very, very deep databases of what human diversity looks like and where in the world it is, genetic diversity, because this is where you get sequenced, where you get genotyped if you would like to be a bone marrow donor, where you have to match genetically, and we know all over the world kind of what people look like here. And we kind of know what Neanderthals look like here too, and it turns out that the Neanderthal sequences exist in humans, and this scale, as you can see, this is for the Denisova. Blue means it's absent, and the Denisova HLA-AC haplotypes are not present in Africa and are present. There are two different ways we can phase this data, two different options for how it could be. Either way, it's not present in Africa, and this phasing, which we think is right, says that this particular set of genes probably has its highest incidence in people today who live in Papua New Guinea, and that close to 20% right around here in Papua New Guinea of all of the variants of this gene came from that Denisova. It's very, very common. Likewise, for the Neanderthal, there are HLA-A and C, different ways you can phase this. And we don't know which one is right, but either way, it's not in Africa. The HLA-B that came from Neanderthal, it's very common in Scandinavia and the British Isles. And the other one is very prevalent, oddly enough, right around Croatia, where we found these bones, which is kind of weird. But appears also to less extent in Africa, which may have been back migration, or it could have been that this is just very common and it happened to be in Neanderthals and in humans today. We don't know what particular pathogen these variants are recognizing, but we would love to know that. What was it that Neanderthals could recognize in their environment and need to fight off that people today who live in certain places might be recognizing and needing to fight off? Why should these variants have come to such high frequency, indicating that maybe they confer some advantage? Okay, so I'll just finish here, and I don't know if I'm over how I'm doing on time. Are you guys bored yet? Okay, I'm a little bit over on time, but I'll wrap it up very fast. So the last thing that I would just like to say is that we want to use the Neanderthal genome to find regions where we have undergone positive selection, things that separate us even from Neanderthals. And having the Neanderthal genome is very useful for this, as you might imagine, because the humans are so closely related to Neanderthals. The variation that we have in humans today oftentimes falls within the variation that existed within Neanderthals. Okay? And to put this in a little more concrete terms, if you go back in time to when the common ancestor of humans and Neanderthals lived, that was about 300,000 years ago. Okay? So I told you the average genomic divergence is about 800,000 years ago. That's how long in time there was a common ancestor at any particular part of the genome, but that's not when the common ancestor population was. The difference between those two, I think, is also illustrated by, if you think about you and your neighbor, you don't have the same DNA sequence. You have a common ancestor for any gene that you might care to think about. That common ancestor for humans lived about 450,000 years ago. So you and your neighbor pick a random place in your genome and ask, when did we have a common ancestor? That's about 450,000 years ago on average. Okay? But the common ancestor population, the group of individuals who would lead to humans and lead to Neanderthals, they lived about 300,000 years ago, more recently than the genetic divergence that is existing within us. This group, we might call them Homo heidelbergensis or Homo erectus. This group, it wasn't one individual, it was a population. And those individuals had genetic differences. And those genetic differences are still alive today within us, and we're still alive within Neanderthals until the time that they went extinct. So there are places in the genome, even without admixture or anything exotic like that, where if you pick a person, they may be more closely related to a Neanderthal than they are to another person. Okay? And if you pick somewhere else in the genome, because of this random nature, they'll be more closely related to a person, and that other person will be more closely related to a Neanderthal. Okay? But this helps us because now we can look for regions in the genome where that's not the case, where everybody's more closely related to a human than they are to a Neanderthal. And these places where all humans are more closely related to other humans are candidates for some advantage that separates us even from Neanderthals. And why do we think there may be such places? Well, we can find the technology that Neanderthals have. This is a very typical technology for a Neanderthal, a Mousterian hand axe. They love to make these. They were very good at them. They're a useful tool. You can deflesh animals, make weapons with them. If you could talk to a great ape and ask him, what do you think about that stone tool there? The ape, if they could talk, they would say, that's amazing. I could never do anything like that. That's a really awesome tool. And it is a pretty awesome tool. But if we compare it to things that our ancestors were doing, even our ancestors that were contemporary largely with the Neanderthals, making things that we can, with our eyes and our brains, look at, and in a few seconds we recognize this as art. We're communicating with the brain of the person who made this. There's reason to think that maybe cognitively we were a little different than the Neanderthals. And if we compare their technology with our technology, especially this plane, which is, I'm happy to say, finally flying, there's even more reason to believe that perhaps we are different than they were. So the idea is we take genetic variation, places where two humans differ, get big catalogs of these. We have enormous catalogs of them now, and just label them. This is some place where two humans may differ. We know that these differences largely come from a common ancestor population that lived about 300,000 years ago. And as Neanderthals went down through the generations, some of these would go up in frequency so that 20% went to 40% of the people had it, or 30% went down to 10, whatever. But largely, what they had when they died came from this common ancestor population. Same thing within humans, unless there's some new variation that comes up that is advantageous. And under this scenario, what happens is something that we call a hard sweep in genetics. If there's something that's so important that you die if you don't have it, or so important that you leave so many more descendants than everyone else, then everything else goes away. And there is no variation around that new variant. If everyone has the same thing, if through a few generations everything else that's different from that one better thing is gone, then there's no variation at all. If we could go back in time, if this happens, if we could go back and get DNA from that time period, we would see, oh my God, there's no variation here. In fact, we see exactly this down here in people today. For example, in around the gene that codes for lactase, digesting milk sugar. If you look in the genomes of people who live in Northwest Europe, primarily, and its frequency goes down from there. There is hardly any variation, and this is dated to about 7,000 years ago when a new variant came up that kept lactase on into adulthood so you could drink milk and not die, get vitamin D and not die. Many, many thousands of people died because they could not digest milk and they lived in this high latitude and they couldn't get vitamin D. You can see this today, but the signal goes away very fast. If we come back in 30 or 40,000 years, we won't be able to see this. And these very old things that happen, we wouldn't be expected to see, and because new variation comes up and you don't see that that was free of variation at some point in the past. But if you compare it to the Neanderthal, these variants now, the Neanderthal never has it because they don't come from that common ancestor. They're newer than that. And in this region, if everything is newer than what we see in Neanderthal, then at some point in the past, the slate was wiped free and something went through human ancestors, something that was new and better. And we've run this and have a lot of evidence that these evolutionary storms happen, but very little understanding of why and what. And that's kind of the description of where we are now. I'll tell you about one gene that we do maybe have some idea what's going on. This gene, we know kind of what it does because there are people who have one copy inactivated and they get something called cleidocranial dysplasia. If you have two copies inactivated, it's lethal and you won't be alive. But if you have one copy inactivated, you get this CCD. And this is characterized by these things listed here. The cranial sutures are delayed in closing. The clavicles have abnormalities. The rib cage is bell-shaped, and oftentimes you get crazy things wrong with the teeth. Supernumerary teeth, more and more sets of teeth just keep coming in. So frontal bossing looks like this. It's kind of a protrusion of the face. Frontal bossing is interesting from the context of Neanderthals, because if we saw a Neanderthal today, we might say they have frontal bossing. They had a protrusion of the face. The forehead stuck out a little bit. The rib shape, bell cage rib shape that you see in people with CCD, X-ray shown here, is eerily reminiscent of one of the differences that we can say existed between humans and Neanderthals. Neanderthals had a bell-shaped rib cage compared to humans. So this gene, where we see evidence of positive selection sometime in our past, if you now inactivate by genetic mutation one copy of it, it kind of recapitulates the Neanderthal phenotype in some way, which gives some clue to perhaps what the advantageous thing may have been. Maybe this morphology was a benefit to us in our evolutionary past. This is an example where we think we have a clue of what's going on. This is very much an exception to the rule where we have no clue what's going on. There's a gene, and you never would have guessed that that gene would have had some important change or even know what that gene necessarily does. But that's what I hope to do over the next 10 or 20 years is get some clarity on that. Okay, so I'll just finish there, and sorry for going over. If you're ever in Santa Cruz, please look me up, and thank you for your attention.
All right, we have time for a few questions. Raise your hand. I'll come by with a microphone. Oh, okay. Just be sure to hold the microphone close to your mouth.
Thank you. Hi.
Hi.
Have you found any evidence that autism might possibly be dated back to the Neanderthals?
Nothing to report. Autism is a interesting, I guess, epidemic, you could even say. It's being diagnosed more and more, and it's something that people are terribly interested in. It's a, putting that aside, the tragedy of it, it's a very interesting condition. Autism is something that's characterized by the inability to do social interaction correctly. And social interaction is something that we think of as very uniquely human. If you could run this autism battery on great apes or other animals, you might diagnose the rest of the animal kingdom as being autistic. We can look in one another's eyes. We do this regularly, and we communicate a lot by looking at through one another. It's one of the things that people who have autism is diagnostic of being autistic, your inability to do that. Other animals mostly don't do that. The only other animal that's really good at this is a domestic dog, oddly enough, and that's probably something that we that bred into it. But humans are very good at that unless you have autism. And the rest of the animal kingdom is also not so good at that. So these kind of uniquely human things that are at the core of being uniquely human are what kind of defines the autism spectrum also. So we're very interested in that with respect to Neanderthals. Are some of these recent evolutionary adaptations, and maybe even something, this is very speculative, that has not come into homeostasis genetically, that very recently changed, does this underlie at all the prevalence of autism that we see today? And I have rambled a lot about this now, but the answer is there's nothing really to report. Yes.
Hi, I was here last week during the Johansson lecture, and I asked him a question about what exactly made us different from Neanderthals, and he actually told me to come and ask you.
Ah.
So, so
Thanks,
as I was
Johansson.
listening to your lecture, there were two things that I thought were, going to make it different, but it didn't. First was the genetic information, and I think the data set is still too small to jump to a conclusion. And then second, second is the skull that you showed us, and you said that it showed that it was clearly different from modern humans. That was the Laferisi skull, I believe.
Laferisi,
but when
yes.
you look at a different Neanderthal skull, like Shanidar, for example, that one looks just like a modern human. It's not as extreme as the one as you showed us. So I don't know, could you put me to rest and tell me exactly what makes us different from Neanderthals?
Probably not. The Neanderthals, like humans, have variance in almost any morphological characteristic that one could measure. And there's a field of called geometrical morphometrics where you measure gazillion different things in a skull, like the difference between these 2 points and these 2 points, and how wide the nose is, and the distance between the eyes and the nose, and just a gazillion different things. And then you find which of these differences are most characteristic of humans versus what we would call Neanderthals. And in these multi-dimensional plots, you can draw a line between humans and Neanderthals. So a precise answer to your question, what makes humans different than Neanderthals, would be some separating hyperplane in this multi-dimensional space of morphological characteristics. But that's not something that that you can really take home or feel like, oh yeah, obviously that's what makes us different than Neanderthals. Another thing that I can say, I think the genetics, I don't think at all it's too early to say that they're differentiable from humans. They're clearly differentiable from humans. This is the genetic divergence between the Vindia Neanderthal and this Denisova relative. On average, this is like 12.7%. We never, ever see any human whose average divergence to another human is that high. This is off the range. So all of these other guys, they peak out at most at about 9%. Nobody goes close to this 12%. So genetically, they are more different than any human, than any other two humans that you could pick. So genetically, the difference between Neanderthals and humans is outside the range of genetic differences between two humans.
Hi. Since Neanderthals and humans mated, wouldn't their offspring be our closest descendant?
If you would like to call this another thing, and but then one gets into kind of a semantic issue with these labels. Is the hybrid offspring of a human and Neanderthal a human, or is it a Neanderthal? And if this is some third thing, Neander human, or whatever one would like to call it, and then this back crosses to a human or back crosses to a Neanderthal. Is that yet a fourth thing? And I don't know the answer to that. You could argue that yes, they deserve their own labels or don't deserve their own labels, but that population of hybrids that have some genetic material from one group and some from the other group, that is non-Africans who are alive today still. It's just the Neanderthal component is very, very small. Yes.
This is a subset of the previous question. You talked a lot about the genes we got from Neanderthals, but if we're interbreeding, don't the gene flow both ways? And so what about the genes we gave to the Neanderthals?
Yeah, that's a very interesting question. We didn't see any evidence for gene flow in the other direction from humans into the Neanderthals that we sequenced here. But there's an important caveat there. We have genome sequence from lots of humans who are alive today, and we see this ancestry in some humans and not others. We don't have genome sequence from lots of Neanderthals. Maybe there are other late Neanderthals that would have human genes in them if we could only find those bones and get that DNA. We only have lots of data from those 3 female Neanderthals from that one cave in Croatia. And in those individuals, we don't see evidence of human DNA. Those are about 40,000 years old, and it's unlikely that humans had made it that far north and that far west by 40,000 years. They probably had already admixed in the Middle East, but probably hadn't made it there yet. So they would be very poor candidate Neanderthals for showing this. Maybe there are some other Neanderthals somewhere else who do show it. I wouldn't be surprised if that was discovered at some point in the future.
We have time for one more question. Anybody? All right. Yes, sir.